За меня же, по-моему, через 2 минуты. Good morning, everybody. I'm glad to see you here. And today we have the second lecture of Professor Batrakova. Again, very interesting topic. Last time she was talking about cell-mediated drug delivery, and today she will be talking about extracellular vesicles as drug delivery vehicles, which is also very interesting and like very fast developing topic in the world. Many papers appearing now, and all of them, all of them cite Yelena's paper and papers already, actually. And this is very unusual to use this extracellular vehicles, because we know that we can add something like liposomes or particles to our body to deliver some drugs. But we can use our own something to deliver them drugs to our body. So this is extremely interesting. And please, Lena, can you get this? Okay. Okay. Happy to see you guys. Good morning. Everybody is listening very carefully. I will ask questions after that. Maybe in the middle, but for sure after that. Or you can have some questions. Prepare some questions. Let's talk. Let's communicate. Okay. Okay. So they can hear me, right? What? Can you hear me?
Yes. Yes.
Yeah,
I can hear. Yes. Yes. Everything is good. Okay. Great. So let's talk about new nature-prepared drug delivery system. So I understood that some of you are working with the nanoparticles, synthetic nanoparticles, and you know a lot about engineering of nanoparticles, looking how they accumulate in the cells and how they deliver therapeutics to the target cell. But today we will talk about, I would say, very promising nanocarriers, which are exosomes. And usually I have some subtitle. One is the official title and another is subtitle. So my subtitle is One Man's Trash, Another Man's Treasure. And this is the old saying again. And I will tell you why I choose this subtitle for this particular lecture. So as objective for this lecture, first, we will talk about what is it, exosomes. So their biogenesis, how they appear to nature, their functions, and then we will talk about how we can use these natural nanoparticles to load drugs in them and to deliver them to the different organs. And as like I told you last time, our drug delivery systems are very necessary when we want to deliver something across impermeable barriers. So we usually working with the neurodegenerative barriers when you need to deliver drug across blood-brain barriers. So, for instance, we specifically, like I told you, focusing on Parkinson's disease. Also, with the lysosomal storage diseases, I will tell you a little bit about that. And about cancer, different cancer, like pulmonary metastatic cancer or triple negative breast cancer. And finally, like everybody asks you, so what? You're playing, you're just working in your lab. How it can be translated to the clinic. So we will talk a little bit about clinical applications and how these formulations already moving from the bench to the clinical trial. So first of all, like I promised you, introduction. So exosomes are fantastic nanoparticles. They produced by almost all cells in the body. When it started, you can read papers and they produced by macrophages, white blood cells. Now they said all cells, every type of cell produce exosomes. And they are kind of new modality, new object, because it's only in 1946, you see, less than 100 years. There was no idea about exosomes or some particles which produced in the body by the different types of cells. And what happened in this 1946, a young female lady, administered herself to the hospital. That was a Columbia University hospital. And she had some troubles with the bleeding. Bleeding was not stopped in her body. So there was two researchers, Erwin Chartgraf and Randolph West. And they tried to study her blood and figure out what's wrong with her blood, why she can't stop bleeding. And they actually discovered that in her plasma, when they just precipitate all cells, that in her plasma, there is some kind of particle. They called it like debris or something, but they figure out that if you spin down these particles and eliminate it, that's when these troubles occur. So these particles kind of help to stop bleeding, to make a thrombus. And it was in 1946, right? So 20 years later, about 20 years later, the Peter Wolf, he actually was a student first at this in these two professors. He actually understands that this coagulation happens because of the platelets. And these are small particles which produce thrombocytes by thrombocytes. And in this case, that was actually extracellular vesicles or exosomes. Nobody knew that they will be called like exosomes or extracellular vesicles. They call it platelet dust. So that was in 1967, let's say how much? Only 60 years ago. There was only some kind of mention or idea that there is something in the blood, some particles that carry some function. So that, in this case, we kind of start this history of exosomal research. So what are the exosomes? These are just fantastic particles. So just a second, I just went too fast. Yes. So I told you all cells release these exosomes. They're about 40 to 100 nanometers in diameter. And they have lipid bilayer. Imagine there's all cells consist of membranes, right? So what they can produce? They produce these bubbles, spheres, which consist of the same lipids, but they also filled with the protein, kind of vectors or targets which can target the cells, these exosomes to target cells, and in some cases direct them to the address which they need to deliver in a message. They are metabolically inactive. I told you last time that cells are promising and very potent drug delivery vehicles, but you need to, respect their nature. You need to have them alive. In case of exosomes, they're just bubbles consisting of the lipid bilayers. So you can isolate them, lyophilize, make a big batch, and then administer to the patient. So it's much easier in this case to use them in clinic. And as I told you, they released by most of the cells, or all cells now, it says. And there all could be B cells, dendritic cells, endothelial cells, cancer cells. But any cell, they talk by means of these exosomes, deliver their messages to their target address. And because they take a lot from the cytoplasm of this parent cell, they retain some of characteristics of this parent cell. So we can make them biologically active. It's not like you use polymer nanoparticle or even liposome. I know that some of you working with liposomes, but when you isolate exosomes from the cells, they carry some characteristics, properties from their parental cells. So you can make them angry, toxic from the angry parent cells, or restorative, nurturing, like, kids. Sometimes they reflect characteristics of their parents. So let's talk about what their function in nature. So like I told you, they are letters. So they enable long distance intracellular communication. And before this idea about exosomes, there was understanding that cells release proteins or hormones or any other mRNAs, any other messages just released into the blood. And these substances travel in the blood, reach their targets, and produce their effect in nature, in a normal body. But then we understand, the researchers understand that it's not that easy. Actually, cells pack their messages, mRNA, microRNA, proteins, into these letters, into the bubbles, into these vehicles. And these vehicles have some receptors that they can travel to specific organs or specific tissues where they're supposed to go. So they have these messages inside. Sometimes it's good, like I told you, we can add some biological activity in addition to the therapeutic substances which we load into this exosome. Sometimes it's not good. For instance, if you isolate these exosomes from the person with the Parkinson's disease, Alzheimer's disease, they actually could carry some wrong messages. And this is a problem. Sometimes we want to clean them up, wash them up, that they will not deliver some unnecessary or unhealthy messages with our therapeutic substances. So I told you that nature is amazingly potent and resourceful engineer. So these exosomes equipped with the adhesive proteins like hooks on their surface. Along with the targeting moieties, they all fill their membranes filled with this adhesive proteins, tetraspanins, integrins, so they could easy to stick to the target cell's membrane. And as you know, like oil, this gets into the oil and water. In this case, these are cellular membranes. So obviously, when they stick, then attach to the membranes of the target cells, they easily fuse with this membrane. And there is about two or maybe more, but we realize two mechanisms how these exosomes enter the target cell. They can fuse with the cellular membrane, the whole first membrane they met, or they can enter by endocytosis and then fuse with endocytic membranes and release their messages, substances, target proteins to the cytoplasm. So they actually have this specific characteristics, this tetraspanins and integrins, adhesive proteins on their surface that facilitate membrane interaction and fusion. And finally, they can also have some ligands. They will target it to the surface of the target cells, like exosomes from macrophages. They will have CD11B from mouse macrophages or CD18 from human macrophages. So everything, and I am absolutely positive that it's not only this mechanism. We usually have like iceberg. We have maybe one of the 100 other mechanisms. We see it on the surface, but there's a lot, very many double, triple mechanisms which facilitate this delivery, interaction with the target cells, and efficient delivery of these messages, mRNAs, microRNAs, or proteins to the target cells. And that's what I'm telling you. This is natural function. So I'm telling you what they're doing in the normal, healthy body. Now I will tell you how they how they appear. Very interesting story. Actually, what I told you about, these are exosomes. Look at this slide. So exosomes produced by multivesicular bodies. So imagine that this is just cells, okay? And it starts kind of invagination inside. And from the cytoplasm, there's make these bubbles inside this multivesicular body. And it's inside this multivesicular body. What they have? They have something from cytoplasm of the cells, right? Now, when this multivesicular body finally constructed, it moves to the surface and releases these exosomes to the surrounding media. This how these exosomes appear. So this, obviously, they are small particles, like I told you, from 40 to 100 nanometers, and they produce by invagination from endosomal membrane to create multivesicular body. Now, there is another extracellular vesicles, and sometimes we use mixture of exosomes and microvesicles. So microvesicles, they're very similar or look like, but their biogenesis is a little bit different. So they butt out from the cellular membrane, plasma membrane, from the big first membrane of the cell. So obviously, they also consist of the membrane bilayers. They also have this adhesion proteins. but they kind of bigger. They from 100 to 500 nanometers, and they have little bit different proteins on their surface, not completely different. A lot of proteins in exosomes and microvesicles overlap, but they have some proteins which are not present in exosomes. So exosomes, 40 to 100 nanometers from the multivesicular bodies, and microvesicles, 100 to 500 nanometers, but at directly from the plasma membrane. And the third type of nanoparticles, of these particles, they're already almost not nanoparticles, are apoptotic vesicles. So we don't use them because they produce when cells start dying by apoptosis. And in this case, it's just kind of shrink and then broken in the many different sizes of these vesicles, but mostly they're bigger than exosomes and microvesicles from 500 to 1,000 nanometers. And basically it's pieces of the cell. And you understand that if they're lipid bilayers, they're never swimming in the water solution like a flat bilayer. They always try to hide their hydrophobic lipid chains inside. So they also produce these bubbles or spheres, but they have a lot of this toxic substances that happen when cells going to die. So we try to use not to use these apoptotic vesicles. We usually, and you see they're very similar, exosomes and microvesicles. So sometimes we think, are you sure you want to use only extracellular vesicles? It's a lot of effort to isolate them from microvesicles, but basically they're very close. So in many cases, researchers use like mixture or together exosomes and microvesicles as drug delivery vehicles. So you remember I told you that this double title, subtitle, when one man's trash, another man's treasure. And what I meant by this, that first idea, what the function of exosome was, these are trash bags. Trash bags. When cell doesn't need anything like some proteins or substance which it doesn't want to, utilize, it just pack into these bubbles and release it. So waste removal or removing unneeded membrane proteins was recognized as the first, and maybe first, they think that it's just main function. And it's still function for exosomes, waste removal, trash bag. But actually later, when people start learn what they are doing, what's their function, they realize that waste removal is only small portion or part of this function. Actually, these are cell-to-cell communicators, ladders, and their function is to deliver one message from the one organ or distant tissues to another organ. And not only distant tissues, these exosomes present even in synapses when neurons interact with each other. And all these messages packed into exosomes and went from one neuron part to another in the synapses. So long distance communicator and short distance communicator, both. And this is a very important function for this nanoparticles. And that's what we want to use, right? That's what we want to use. We try to understand how nature communicates, and we try to use it to deliver our drug to the distant organ or to organs which is really difficult to reach. It's also very important function as an immune modulating property. I will tell you about it later. But actually, when body recognizes that you need to protect or restore or fight with the, let's say, cancer or other other diseases, exosomes from different cells fighting with that. They deliver these messages. They show this is the bad. Look for immune system, and T cells recognize these parts of the proteins on the surface of this exosome, then move to the B cells, to lymphocytes, and teach them how to, which kind of antibodies you need to produce to and finally, a huge part of this function is protective and regenerative effects. So you can find thousands and thousands manuscripts, reports, research papers that would tell you the exosomes from endothelial cells help cardiomyocytes, cells of the heart to restore, help cells from the liver to survive, help neurons in the brain to fight with the degeneration. So a lot of the functions of the exosomes from different organs to protect and regenerate. And as I told you before, exosomes basically, think about them as a naturally prepared ladders. They know where to go. They have their address on their surface in these receptors which they express. Cells equip them to find where to go. They can efficiently attach and deliver their message to the target cell. But like there happens with the senders. Senders could be good, it could be bad. So exosomes as a ladder can carry good or bad messages. For instance, cancer cells produce a lot of exosomes, and they equip them with a very toxic and basically scary function. Exosomes from cancer cells were shown to promote oncogenesis, start new metastases in different distant organs, suppress immune response. Can you imagine? They just teach these T cells and macrophages to close their eyes and to not to fight with the new cancer cells. And they even can deliver proteins that work as a for drug resistance development, like PGP, MRP. So it was shown that these exosomes can, from cancer cells, can deliver a lot of bad functions. But it's like with the senders. If there's a good sender, he will send a good message with restorative, immune modulating properties. If it's a bad sender, like a cancer cell, they will deliver a lot of very unhealthy, toxic, and dangerous properties. So now let's talk a little bit about intrinsic biological activity of exosomes or extracellular vesicles. So exosomes, one part, right? Like I told you, exosomes and microvesicles. Together, we call them extracellular vesicles. So a long time ago, there was an idea. Let's try to teach cells of the body to react on the cancer. For instance, patients get to the hospital. They take the tumor out. And then the idea was, let's chop this tumor cell and inject back. And they will show the immune system that this is bad, and they will activate immune system, and the body itself will fight against new metastasis or development of this tumor if it's not resected completely. But later it was found out that it's not good. These chopped cancer cells, how much you can chop still, they can produce new tumors. So there was another idea. Let's take these cancer cells and expose them to the immune cells of the body outside, ex vivo, in some plate or tube. So these dendritic cells or macrophages, all immunocytes, they will look at these cancer cells, activate, then you isolate this activated immunocyte and inject back to the patient. Now you just injected these immunocytes which know that fight with this particular cancer. Didn't work too, because it's just this immunocyte could deliver some parts, and this cell injection also was used, but not that efficiently. But finally, they figure out, let's collect exosomes from these dendritic cells. So first you expose this tumor tissue to the immunocytes, activate them, wash it out, and then you keep these cells in the media for 2-3 days, collect exosomes from this immunocyte. And these exosomes will also carry this message, fight with this protein, and you can inject this into the patient. So that could be used as anti-tumor response, triggering anti-tumor response for the patient. And like I told you, protective and regenerative effect. Specifically, look, it's a huge list, and you can find more and more properties that these extracellular vesicles from mesenchymal stem cells can deliver. So you remember yesterday I told you about, last time, about mesenchymal stem cells that produce, you can isolate them from bone marrow or from the adipose tissue. It's just like fat, from the fat tissue from patients, cord blood, or other origin. So you have this mesenchymal stem cells, culture them in the plate, and then collect exosomes from them. And these are very potent and powerful and positive cells, which can protect, give cardioprotective effects, like protect cells of the heart when it was heart attack. It also have anti-inflammatory and anti-oxidative effects, anti-apoptotic, anti-fibrotic, stop this plaques appearance, and cardiomyogenic effects, even restore cells of the heart when it was after heart attack. It's also working against atherosclerosis, cleaning vessels from cholesterol on this deposition. It was shown have neuroprotective effects in case of stroke. It's in the brain, right? Insult, stroke, Alzheimer's disease, Parkinson's disease. It's these exosomes from mesenchymal stem cells could have this neuroprotective effect. And hepatic regeneration. This is the liver. So a lot of cirrhosis now can be treated and improve the liver cells hepatocytes by using these mesenchymal stem cells derived extracellular vesicles. So these are a lot of, that's what I'm talking about, biological activity of these carriers itself that we can use to add this activity to the drug which we incorporate in the cell. So using of these exosomes or extracellular vesicles as a drug delivery vehicle has a lot of advantages. First, they're very stable, much more stable than let's say, liposomes from lipids, prepared from lipids. Second, they're biocompatible. They're the particles from the same body. So they have low toxicity. And like I told you, they have intrinsic biological activity. They have this targeting receptors like ICOM1 or LFA1 receptors that they can target to the inflamed tissues and to the cancer cells. Like I told you last time about macrophages, they can be targeted to inflamed brain tissues, inflamed brain endothelial cells, the blood vessels in the brain. The same property has this extracellular vesicles. They kind of get these properties from their parent cells. They also can accomplish efficient drug transfer. They have adhesive proteins, and they have these proteins involved in the trafficking and membrane fusion. It's all natural mechanisms, powerful. It's amazing mechanism that all body prepared to communicate between the organs. There is also possible that we can add some vectors, grafting, targeting molecules on their surface. They have low immunogenicity. This is another very interesting story. The extracellular vesicles equipped with specific proteins like CD47 or CD49 that that these all immunocytes, phagocytes, white blood cells, they target particles which look like bacteria or viruses, 100 nanometers, 200 nanometers, accumulated very efficiently, like we talked with you on the last lecture. But these very important particles equipped with this CD47, 49 receptors, and they act like they stop accumulation in these phagocytes. So when macrophage or dendritic cells try to engulf, take this particle inside, it interacts with this CD47 and stop, open the mouth and let this particle go. And this is very important property because all macrophages, all phagocytes just looking for nanoparticles because usually in nature it's bacteria or viruses. But if body tries to send a letter to another organ, it will take care that letter will be protected from this elimination by phagocytes. And so that's called "don't eat me" effect. So it's like with this fish, which the small fish, the story, just release some substances and it frees the mouth of the big fish and can fly away like flounder. So same here, don't eat me effect. So these proteins which express on the surface of this exosome, they stop accumulation in phagocytes. So basically, you have invisibility clock. It's like they release these particles, but they are invisible to the immune system. They are not toxic. They know where to go. They can deliver efficiently, fuse, produce effect. Fantastic. Fantastic. We only can use these natural mechanisms, many mechanisms. Just need to load our drug and then it will go and work by itself. So the second part of my lecture is how. That's not easy. And we are not that practitioned and resourceful like nature, but we are working on it, and we learn that there could be three different mechanisms or ways how to load drug into this exosome. First way, and I told you a little bit about this in our previous lecture. First way, transfect parent cell with the DNA that's encoding therapeutic protein. It's usually for proteins, right? So in this case, we will take macrophage or dendritic cells, mesenchymal stem cells, whatever cell you want to use with this encoding DNA, and then collect exosomes. And as I told you last time, you remember, this exosome will have not only encoding protein, but also DNA, plasmid, pDNA, and mRNA, and even transcription factor. So very powerful kit for transfection tissues. So this is the first way or approach how to load exosomes in with the drug. The second approach, again, I told you about this a little bit last time. You put nanoparticles, you remember nanozyme? You load it into the parent cell, and then you collect media, because in this media, these exosomes released by this preloaded parent cell will carry this nanoparticle with the drug. Actually, we will not talk a lot about these two ways, little bit, because I told you about it last time. But just to remind you, you can transfect cells and collect exosomes. You can load them with nanoparticles to protect drug inside and then collect exosomes. But the last one, we will focus in this lecture because I don't want to repeat you what I did last time. So the third way is you collect naive exosomes without any drug. And then you working with this, they're not empty, right? They have their own messages, but we call it naive or sham exosomes. And you work with them to load them with the drug and then to look how they will deliver and how they produce therapeutic effects. So it's not easy, as usual, but first you need to characterize them, purify them, right, from the media. And there's several methods now, and a lot of companies working to isolate, to produce clean, pure, very well characterized formulation of first empty exosomes or free, naive, sham exosomes. So you can use differential ultracentrifugation. It's when you just have this media and you centrifuge this media at different speeds, and you finally you have pure part of this extracellular vesicle. It's labor intensive, it's time consuming, but it gives you high purity. And basically now we're working mostly with this differential ultracentrifugation method because these extracellular vesicles are well characterized, purified from proteins, from the same media, purified from the apoptotic cells. Another method, quick, cheap, and they're pure, but not that pure. It's polymer-based precipitation. When we started working, we usually use this polymer-based precipitation with polyethylene glycol pack. It's just like this high concentration of pack. You put it in overnight on the tube with exosomes, and it's like a mesh. It take all exosomes and pull it down, and then next overnight, and the next day you spin, and you have this exosomal fraction. We started with that. A lot of proteins accumulate together with exosomes, so later we decide we better use differential ultracentrifugation. Another method, even more pure but more labor intensive, is immunoaffinity chromatography. You can imagine. So it works on the proteins which exposed on the surface of this exosome. So you can buy some magnet nanoparticles which will have these antibodies to this specific protein. So when you mix them with the media, exosomes will stick to these particles, and then with magnets, you isolate together, and then with the high salt, you take this highly purified fraction of exosomes. And here you can, for instance, separate exosomes from microvesicles or purified exosomes from one cell. You remember I told you last time, like T cells, one receptor, macrophages, another receptor. So you can highly purify it. You have very low, quantity of that because you purify them from all others. You lose a lot, but you have very high specificity because of this isolation. And finally, size exclusion chromatography. This is highly reproducible and sensitive method, and it preserves integrity of exosomes, because when you work with this precipitation with the magnetic particles, sometimes you can break this exosome. So there are different methods, and depending on you want more or more pure, you can choose the method for isolating these extracellular vesicles from cell media. Now you isolate these particles. Now you need to characterize. So there are several methods we learn, and it's very important to characterize them. So you can characterize them by size, shape, and morphology first. So we use nanoparticle tracking analysis, or dynamic light scattering, or atomic force microscopy. You see, you even can see these particles, or transmission microscopy. You can just see these particles, and even sometimes with the drug inside. Next, you can characterize them protein and lipid concepts. So we can use Western blot to see, or by flow cytometry. You can use mass spectrometry or microscopy techniques just to characterize by the protein. And they have specific proteins on the surface of exosomes, for instance, TSG101, Alex, Flotilin 1, and others. We always, when we publish papers, we have to characterize them first. And they actually have some lipids, specific lipids on this exosome, like single myelin lipid, or they enrich with this hexacylceramide, difficult to even pronounce, but they're specific lipids, and you can characterize them by lipid concept. So there's what about characterization. And if you think that it's just in dreams or something which is not real, it's not true. There's a lot of researchers which load now a lot of different drugs. And first, of course, a lot of efforts to treat cancer. So low molecular drugs such as doxorubicin or paclitaxel, or antioxidants, also curcumin, the low molecular drug, they were loaded and reported a lot for exosomes. There's also now a huge effort to load nucleic acids. So siRNA, mRNA, DNA, all was exosomes all used for loading this drug and used as a drug carriers for this nucleic acid. In proteins and peptides, for instance, we use catalase redox enzymes, also sometimes very big enzymes we use to load into exosomes. And if you will just consider if it's hydrophobic agent like paclitaxel, it will be incorporated into lipid bilayer, right? If it's hydrophilic, it will probably go inside this vesicle. So you just need to consider how much you can load, because actually these particles has their own proteins and microRNAs. Or this lipid bilayer also kind of limited. You cannot load it for forever for a huge amount. So that, but efficiency of delivery is high. So this is why we want to use these exosomes, which are non-toxic, but can be targeted and efficiently deliver the drug. Now, different approaches were used to load exosomes. Obviously, I don't want you to memorize this table. I just want to show you effort, how many, different reports about how you can load exosomes. For different diseases, look at that. For cancer, brain diseases, for, for lung cancer, for a lot of different diseases, neurodegenerative diseases, Parkinson's disease, prostate cancer. They use a lot of exosomes, different source of exosomes. They bring a lot of different cargo. Like I told you, paclitaxel, doxorubicin. I will tell you about TPP1 for lysosomal storage diseases, sRNA, catalase, and the loading. Loading, most of them started with co-incubation. You just drug, mix with exosomes, and look how it would work. Try to load, but it's not very efficient. You need to make this lipid bilayers permeable. They are kind of, strong and good density. So we use different methods to make kind of holes in these exosomes, like sonication, electroporation. And so that's what we're doing. And first, I will tell you about Parkinson's disease. We talked about it yesterday, last time, a little bit, but you remember that you want to deliver drug to the substantia nigra. You remember this kind of like two like wings, black dopaminergic neurons. They localized in the midbrain or substantia nigra, black part of the little bit black tissue. So it's you want to deliver this drug to this substantia nigra. And Parkinson's disease first discovered in 1817. He has this patients, he called them paralytic agitants. He looked and he described their problems in the essay on the shaking palsy. And he showed that they have a decreased spontaneous movement, they have gait difficulty, postural instability, rigidity, and tremor. So he has no idea why and how it happened, just described this, and his name was Parkinson. So since then, we have the name for this disease, Parkinson's disease. And we use the two most important therapeutic agents, and I told you about them last time when we use cells as a drug delivery vehicle. Here we try to use exosomes from the cells to deliver these two most powerful therapeutic proteins. It's a glial cell-derived neurotrophic factor. You remember, it protects neurons and make regeneration. And the second is catalase. The most potent antioxidant is just stop inflammation and prevent degeneration of neurons. Not reverse, but at least prevent. And now I will tell you about what we're doing to load these exosomes efficiently. So we collect exosomes we work with macrophages, from macrophages, the monocytes, And then we try to fluidize membranes, make their membranes more permeable for drug. We incubate them at room temperature with or without saponin. And saponin is known to make a transient hole in this membrane. So you take this catalase and you mix them with exosomes, and then you add saponin to make holes and let this catalase get into exosomes. Then we use freeze-thaw cycles. You know, freeze-thaw, maybe there's membranes will be disturbed more or less, and we can load this catalase inside, or electroporation. We took this electroporation from the transfection. Usually when people want to transfect cells, they add like DNA or mRNA and electroporate them. And under this electroporation conditions, the membranes start make holes. So DNA or mRNA gets inside the cells. We decided to try the same with exosomes because they're still the same membrane, but not by membrane of the cell, but membrane of this small particle. We also use sonication. It's just ultrasound. You can sonicate them in the water bath, or you can sonicate them with the just sonicator, and it's much more powerful. Or extrusion. We thought that how you make liposome. You push them through the mesh, extrude them, right? Lipid. So let's try the same with exosomes. Maybe when they will get through this mesh, their membranes will, stretch and make some holes, and maybe this extrusion will help us to load this catalase into the exosome. And first we'll look by FM what we get, which kind of particles we get, and look what we found. Wow, these are incubation at room temperature. Nice, like sphere particles. Saponin, we like it. There's again nice sphere exosome. Freeze-thaw, no. Freeze-thaw won't work. What happens when you freeze and thaw many times? These exosomes interact with each other by their adhesive protein. So they clump together. And in this case, you have a not good formulation. It's big, small aggregation. So of course, we could load some drug in between or inside this exosome, but it's so bad formulation, we decide to abandon that, not to use that. Sonication, actually, look at that. Strong sonication destroyed these exosomes. We got some UFOs, like triangles. So unfortunately, strong sonication wouldn't work. But mild sonication, we can use them. We have these particles in this mild sonication. We very much use to load exosomes with different therapeutic proteins. Extrusion works good, but with extrusion, actually, again, when you push it through the mesh, they interact with their adhesive protein, and you lose about 50% on this mesh, on the surface of this filter. So extrusion won't work. So we look first, we said, okay, saponin and maybe mild sonication would work the best. And then we characterize them, like I told you, by size with the catalase, for instance. By size, we looked at that, okay, there's 100 nanometers, good. They can go into the target cells by endocytosis, and by nanoparticle tracking analysis, just compare it. Characterize them by size, and then we characterize them by the amount of catalase loaded into this particle. And we compare it like this 1, 2, 3, 4, 6. So number 3, it's incubation just at room temperature. 4, freeze-thaw. 5, sonication. 6, in extrusion. So you can see more and more catalase was loaded into exosome. And the best, 5 and 6. So sonication and extrusion. We looked at by Western blot, is just how much catalase. Then we looked by enzymatic activity, how much this catalyst can deactivate free radicals, and we looked how it just releases from the cell, from the particles. So again, same like with the cells, the more you load, the longer it gets and release like over 25 hours. By sanitation, you have released only 50% of this catalyst. You need to think about, do you want this release fast or not? It all depends on the formulation. But basically, if these particles keep them longer so they can reach the target site and then release it, overall, it's good. So we choose the sanication extrusion, basically sanication, which is mild sanication, when you don't destroy these particles. And we look how they interact with neurons. You remember beautiful pictures last time? Better one time to see than 100 times to hear. Same here. So these are dark neurons in vitro in the plate. And here is exosomes loaded with fluorescently labeled catalysts. So they are red. They're exosomes and with the catalyst loaded. This not one exosome, this basically aggregates also. So you can see that 10 minutes, there's neurons dark, but these exosomes stick to the neurons with their adhesive protein, and they stick more and more and more, and finally they send these catalase inside neurons. At 75 minutes, you can see already that all neurons are red. There were nothing here, 0 minutes, all neurons are red. What it means? So these exosomes attach to the surface of this neuron, fuse with the membrane of neurons on the with the plasma neuron membrane outside, or even inside with endocytic membrane, and then they deliver this catalase into the neuronal cells. This is in vitro. This is just you plate neurons, add exosomes with fluorescently labeled catalase, see how it goes over time. Now, interestingly, but exosomes obtained by sanication deliver the highest amount than compared to freeze-thaw or incubation at room temperature. Obviously, the more catalyst you load, the better you accumulate in the cells. But actually, this is the how many cell exosomes accumulated. So even if you don't load it with catalyst, but look how fast these exosomes get into the target cells, sanication is the best. It's puzzled us. We tried many times. We still see that sanicated exosomes get into the cells more efficiently. And finally, we hypothesize. It's not like the absolute true, but we hypothesize that when you sanicate, maybe some hydrophobic parts of the lipids get exposed and that maybe they interact better with the cells. A lot of mystery, and we cannot explain everything, but at least we can propose some kind of reason why it happened. And then the maybe very important, or maybe the most important part, we showed that these exosomes from macrophages, same like macrophages, they get their properties, they target inflamed tissues, they target sites with the disease. So we can deliver these exosomes with the drugs specifically to the inflammation, to the disease site. One of the mechanisms, not just only one. There's multiple, it's like an iceberg. But one of them is expression of LFA1 receptor. So it is known that LFA1 receptor interacts with ICOM receptor on the inflamed tissues. All cells where the inflammation produce this ICOM1 protein, and it's like a light or flag for macrophages. They target these ICOM receptors and they slow down at that. You remember last time I showed you this macrophages that roll, slow down, and then get into the brain. Same here, but these are exosomes. So we demonstrated that if you isolate exosomes from macrophages, they express a lot of this LFA1 receptor, and actually they accumulate in the inflamed tissues better than non-inflamed tissues. So at healthy conditions, macrophages and exosomes, they kind of pass by. Yes, they, of course, like I told you last time, they penetrate between cells at the macrophages and through the cells. But exosomes interact mostly with this ICOM-1 receptors. And again, same like with the macrophages, they target inflamed tissues, target and deliver your drug to the site of the disease. Very important. So adhesion, like I told you, with the different proteins that's specific for this exosome, and then fusion with plasma membrane, and it could be with the plasma membrane on the surface of the cell or through endocytosis mediated transfer. So they get, because they're small, less than 200 nanometers, they're able to enter into endocytic vesicles. And then inside there's, for synthetic particles, it's a big, huge problem, endocytic release, because a lot of synthetic particles cannot cross this membrane from endosome to the cytoplasm. And eventually they get to lysosomes and get destroyed by the lysosomal enzymes. But exosomes, naturally prepared messengers, they have their specific ability to interact inside this endosomes, fuse with endosomal membrane, and release their carrier, their content into the cytoplasm. So obviously we want to compare it with the other synthetic nanoparticles. And in this case, we compared accumulation of exosomes with the polymer-based nanoparticles, PLGA nanoparticles. So this column is exosomes accumulated in the neuronal cells, and the second column is PLGA nanoparticles accumulated over 24 hours, first row, and over 72 hours, second row. So you can imagine, we add the same amount of nanoparticles with the same fluorescence. So here and here, there's the same fluorescence was added to this neural cells. But see what's the difference, especially at 72 hours. Look, here is very little amount of PLGA nanoparticles. Basically what happens when cells take these nanoparticles, it's recognized inside that it's something foreign, something which is not necessary for the cell. So it's recycled, get out it. And so 72 hours, you have very little amount of nanoparticles, predominantly in lysosomal compartments. But exosomes, with their adhesive proteins, with their lipid bilayers, they just overpower cells. Look at this neuron. It's just they stick, they go, fuse, deliver their carrier. So it's a huge difference between synthetic nanocarriers and naturally prepared nanocarriers, exosomes. And because it was catalase, it's like we imagine it's like a cat, which is with their adhesive proteins, it's just stick their nails, get into the cell and deliver its drug. Now, if we will go to the in vivo, how we do that. So obviously the same like with the cells, like I told you last time, you want to have the same exosomes from the same type of organism, body, because they also will be immunogenic if they will be from another type of body. So this is actually limitation, like I told you with the cells, limitation. We want to have autologous exosomes from the same body, from the same patient. but there are several ways how to overcome it. But when we work with mice, we first take bone marrow, harvest it with the one mouse just from the bone. Then we grow this bone marrow cells with the stimulation factor, macrophage stimulation factor. They produce mature macrophages in the plate. Then we isolate exosomes from these macrophages media and load them with the drug and inject to the same type of mice. So it's the same strength. So if it's CB57 black, we will work with the CB57 black mice. If it is bulk, we will work with the bulk. And then we look how they go into the brain. So first, we label these exosomes with the fluorescent dye, and then we injected them into the mice. So you remember last time I told you how we produce this inflammation in the brain when we inject toxin into the substantia nigra 6-hydroxydopamine, and then we administer this exosome. So we try first two different methods: intravenous, and this is C here, or intranasal, and this is B. It's part of the brain, or whole brain, also intranasal. So exosomes were labeled with the red fluorescent dye, and blue are nucleus. So we demonstrated that actually there is a significant amount of exosomes in the brain tissues when you administer into mice. But intranasal administration gives better results. So, and interestingly, still I don't understand how it happened. But look, these are like necklace around the nucleus. These exosomes somehow concentrate around the nucleus, and in addition, there's overall tissue staining. So they kind of disperse throughout the brain. So we decide, okay, intranasal administration the best, maybe because they don't stick in the liver and spleen. You remember with macrophages also, this peripheral organs, they're working, there's their function, collect all nanoparticles. So intranasal kind of, they go through the neurons along with these neurons of olfactory in the nose, straight to the brain. And then we even looked where they're going. So we took these brain tissues and we labeled with the cell marker for astrocytes, the red exosomes. These are just DAPI nucleus, and these are cell markers: astrocytes, neurons, microglia, it's residential macrophages, and endothelial cells. So when we label them, we saw colocalization basically with neurons and with microglia. Microglia, it's understandable, they're phagocytes. They will, even with this don't eat me effect, they still will try to accumulate this exosome. And neurons, great, we're working to deliver our drug to the neuron. So we moved further and look how it was effect. Again, you remember this picture. So this is substantia nigra. Neurons specifically stained for dopamine, and they have this kind of shape through the midbrain. These are healthy neurons. These are with the empty exosomes. They don't do much. They don't kill neurons. Great. These are degenerated neurons because we injected the toxin in the brain. And these are the same injected toxin, but with the exosomes with catalase. We showed some protective effect, not just perfect, but still we have a lot of neurons survive after we treated these intoxicated mice with exosomes with catalase through intranasal administration. And then we move to another model, Batten disease. And in this case, you remember I told you different ways how you can load exosomes. We use these two ways. We took the white blood cells or macrophages And in one case, we transfected them with DNA encoding therapeutic protein. Or in another case, we take naive exosomes and loaded them with this therapeutic protein, and then we characterize them in vitro first, how they protect neurons in vitro, and the second, how they protect neurons in mice. So a little bit about Batten disease. Horrible disease. It's a heritage disease. So it's taken by the parents, from the parents, with the genes. So in patients with this disease, it's juvenile disease. So about kids about 12 to 14 years old, they die because there is not enough enzyme that destroy the plaque. And this enzyme is TPP1. So when there is it's not working over years. Some diseases like 5, 6 years old, 1 month patients die, but this 12 years old. So over years, this enzyme is not working, and the aggregate in the cells appeared more and more. And specifically, neurons are the most vulnerable for this. So when this plaque increase and increase, finally neurons start dying. And that's what caused the death. First, kids stop talking, then stop working. Already they were good, they were perfect, and then decline. And at 12 to 14 years old, they die. So the problem is you can deliver this TPP1 to the peripheral organs, but brain is impermeable, blood-brain barrier, difficult to deliver. So what we need, we want to use this exosome. We want to load them with the TPP1, and then we want them, administer them, that they will get into this lysosomal compartment and destroy this aggregate. When they destroy, they protect neurons. Very simple scheme, very difficult to achieve therapeutic efficacy. So first, we look where this exosome is going. And here we have colocalization. So in this case, exosomes are red and lysosomes are green. So we looked, and so in this case, it's nature working for us. So whatever nanoparticles get into the lysosomes, that's what we want. We will deliver this TPP1 in exosomes, and actually a lot of them get finally to lysosomes where these aggregates combine together, and we maybe were able to destroy this aggregation and protect neurons. And then we use mouse models. So these are specific mouse model knockdown mice where this gene that's supposed to produce TPP1 knockdown, there's not working. And in this case, you can treat these mice with your exosomes or other different formulations and see, will it work with these mice or not? They usually die out about 4 or 5 months, 4 or 5. And we looked for how you remember first with the Parkinson's disease, we realized that intranasal administration would work better than intravenous, for instance. In this case, we went further. We tried to check for intravenous administration. This is IV. Showed you for macrophages last time. Intravenous. We tried to do it intraperitoneal. Intrathecal, you remember we liked this intrathecal last time for macrophages. We still want to deliver to the brain, right? And intranasal. The problem here, and like everywhere, is that intravenous you can administer a lot, like 200 microliters into mouth. Intrathecal, like I told you last time, only 50 or sometimes even less. Intranasal, 10. So we demonstrated that, of course, these exosomes A lot of them in the peripheral organs, intravenous, obviously, liver and spleen, but they can reach the brain. And actually, we showed that healthy mice showed less accumulation in the brain, same like with the macrophages. And intrathecal administration, also very promising, because in this case, same like with the cells, you avoid entrapment, accumulation in the peripheral organs, liver and spleen. So we choose two ways. Actually, you need to start treat these animals very early, like day 10 or day 7. They're very small yet. So inject intravenously really, really, really problematic. So we decide to choose this intraperitoneal administration because you just can inject it into the belly and that's it. And then when they grow up a little bit more, we decide to use intrathecal. Same like a cells, we decide maybe it will be more efficient. And that's the survival curve which we found for this experiment. So this first fast died out are saline or extracellular vesicles without TPP1 and or TPP1 alone. So all of them kind of no different. When we injected the process, look, there they live for 100 days. They're healthy mice, not knocked down. They have their own TPP1. Most of us, we don't have problems with this disease. But if we inject intraperitoneally, these are squares, we help these mice to live longer. But especially when we have this combination of intraperitoneal and intrathecal administration, we have very good survival curve, which is we significantly prolong their survival compared to non-treated Batten disease mice. It's not perfect. Unfortunately, we cannot completely eliminate this problem, but at least we are on the right side. We're working further to improve this formulation to show better and better effects. Another very interesting approach is when we use these exosomes for cancer treatment. And remember, I told you that sometimes it's really difficult to reach this metastasis. So we work with these exosomes to try to deliver to the metastasis in case of pulmonary metastasis. And in this case, we, as usual, we take macrophages, we isolate exosomes from this media of these macrophages, and then we load them with paclitaxel or doxorubicin. I will tell you how. It's a very interesting approaches, but it's you need to work on it. It's not happen just you mix it and they load it. No, it's not an easy task. And then you add to the cells, they will target cancer cells through the same LFA1, ICOM1 receptors, and we thought it might help to eliminate this cancer cells. Again, same scheme. We use different ways of loading, like you described you for catalase, but in this case, it's kind of easier compared to catalase. Catalase is huge protein. You don't want to destroy, deactivate this enzyme. Paclitaxel or doxorubicin, they're low molecular drugs, kind of can survive upon different loading conditions, and they're smaller. So that's easier to load at a high quantity. And then we look at how they work with the cells, how they accumulate with the cells. It's just so beautiful. I couldn't resist. I compare the accumulation of exosomes in cancer cells, it's in vitro, in the plate, with accumulation of the same amount of liposomes or polysterol nanoparticles. So this is the quantity, how much. So you can see the huge difference. Yes, it's difficult to produce. Yes, it's more difficult to load, but how more they accumulate in the cells compared to synthetic liposomes or polymer-based nanoparticles. And then we look how they get into the triple negative breast cancer cells. In this case, the problem is that if some breast cancer cells express some proteins, specific proteins, which you can use and fight this cell with the antibodies to this proteins. But in triple negative breast cells, they do not express these three proteins which you can use fighting with this cancer cells. But extracellular vesicles working by their own way, using LFA1 or other adhesive proteins, which we don't know even. So you can see how much doxorubicin and paclitaxel was accumulated in these triple negative breast cancer cells in vitro, in the tube, compared to the same amount of drug loaded into liposomes. And after that, we obviously characterize them. You see, we found these proteins on the surface, which specific for the exosomes. Alex, you remember I told you TSG101 flotillin, characterize them by the size and by the how they load, how can we load these exosomes with the drug. And here, sanication, again, sanication worked the best. So mild sanication, not the sanication when you destroy everything, mild sanication. We characterize, we play with this loading. Actually, we can change the pH for doxorubicin, make it more hydrophobic, pH close to pI. And in this case, we can load it better. We played for the sanication at ice or at room temperature. Interestingly, so when you want to try to load this drug in exosomes, their membranes better be warm, because if you keep them on ice, they're rigid, they're strong, and there's much less loaded drug compared to the warm membrane. So it's art. It's not easy, but it's promising. It's worthy to try to load them and use these exosomes for drug delivery system. We also showed that they actually can overcome resistance of the cells. And we used the compared resistant cells and sensitive cells and showed that probably using their own receptors, they still can accumulate and deliver more drugs in resistant cells compared to other formulation like Taxol. It's also particles, but it's oil-like particles, and they have much less ability to overcome drug resistance. So even more, you remember I told you that we can even graft them with specific vectors to target to the cancer cells. And in this case, we use this aminoacyl anizamide, AA, decorated exosome. Obviously, you have this lipid bilayer, so you can use kind of complex of lipids on one side and then this AA protein on the surface. So you sanicate, load it, and vectorize this exosomes, and then you characterize them. You show that they have this AA receptor, and sigma R is the receptor on the cells. You show that there's a lot of this sigma R receptor on the cancer cells, and aminoacyl anizamide interact with them and increase accumulation in this cell. So theoretically, if you know the vector moiety to the specific your target cell, would it be sigma R or ICOM1 or other receptors, you can use it to improve, increase targeting delivery of this exosome to specific cell types. Here we learn that actually when you load this vector, you kind of decrease the ability to load drugs, you see? So it goes down. So kind of there's not so much space. So you need, again, optimize and find the quantity of vector and that would accumulate a lot of, you can load with the different drugs. And then you can see that they accumulated in the cells very efficiently, and we can deliver them to the pulmonary metastasis. So the problem with pulmonary metastasis, a lot of problems, and it's difficult to target it. For instance, if like breast cancer tumor, you can basically resect it, cut it off. With pulmonary metastasis, you never know where, so many. So it's really difficult to take them from the body. And you need to target your drug to the pulmonary metastasis and kill only cancer cells and not normal tissues. And here we found amazing properties of exosomes. So these are lungs of mice which were injected with the fluorescently labeled tumor cells. So this is a specific model. You take this red tumor cells, inject them intravenously in the mice, and they populate lungs. So after some time, when they develop this metastasis, you take lungs, cut it, and see by fluorescent microscope. You see that that's all lungs with the red cancer cells. Then you inject into this kind of mice exosomes. You label them green just to see where they're going. And can you imagine, they completely colocalize with the cancer cells. They don't care about normal tissues over here. They just target and fuse with the cancer cells. That's amazing. So you can send your cytotoxic agent only to the tumor cells, avoiding treating normal, killing normal cells. These are higher magnification. Even more, it's clear to see. So here is the normal tissues, right? These are cancer cells, red ones. And here, the same slide, but in the green fluorescent light. So you can see that yellow colocalization practically only in the cancer cells. How they do that, who knows? But macrophages just naturally prepared cells to target cancer cells, right? So exosomes probably have this LFA1 or other receptors which target cancer cells. And it means that we can efficiently deliver the drug. And here is the result. The red, again, it's cancer cells. We make this model, and mice treated with saline develop all these metastases. We try to treat this mice with Taxol, common anti-cancer reagent. Didn't work much. We tried to treat them with exosomes with PTX, and it was already very efficient. We saw that these exosomes reached this cancer metastasis and eliminated them. But if you have this vectorized exosome with paclitaxel, you basically completely, at least on this slide, you treat and eliminate this metastasis. And then we looked for a negative breast cancer. I told you there's 3 receptors that usually used to treat this breast cancer cells, but triple negative, they don't have this receptor. So treat this triple negative breast cancer is really difficult. And we made this model in mice. We are still working with mice. When we injected these triple negative breast cancer cells, we make them green. And we injected red exosomes. And look how they colocalize. And here we tried intratumoral. It's not intrathecal here. It's intratumoral into the tumor. intravenous and intraperitoneal administration. Intravenous, probably intraperitoneal the most, because intratumoral, what? You better cut this off. So when you inject intratumoral, obviously there's a lot of exosomes in the tumor, but it's not that important. The important is that when you have intravenous and intraperitoneal, these exosomes colocalize with the triple negative breast cancer cells, this yellow color. If you use the same, but instead of exosomes, we use some other particles, like in this case, it's polymer particles, I believe. There's nothing, only when it's only in the tumor. They do not reach tumors. And here's polystyrol nanoparticles. Before it was liposomes, and here's polystyrol nanoparticles, synthetic nanoparticles. Again, they are not reaching tumors upon intravenous and upon intraperitoneal administration. And if you will go again to the exosomes, huge difference. Intravenous administration, intraperitoneal administration for exosomes. This is for liposomes, and this is for polystyrol nanoparticles. So even in vivo, you can see how big difference makes this using this naturally prepared carriers. And obviously, we significantly decrease slow down tumor growth, not completely. Again, unfortunately, we wish, but unfortunately, this is not 100% elimination. And we're working on it again, suggesting different approaches, but it's still therapeutically significant. So in clinic, we started with you with a huge table. This is another huge table. So this is how this exosome from different cells used for clinic. Can you imagine? This is all mesenchymal stem cells, exosome released by mesenchymal stem cells. We actually also use from plasma, even from plant, and that's what I wanted to tell you. From grape, plant, grape, they also produce exosomes, milk. If you isolate exosomes from milk, tons of exosomes. So you can use other exosomes. They might not work as good as from the same body, autologous exosome. But here's a huge example for you. A lot of examples how this exosomes used in a lot of clinical studies. And there is some description of first clinical studies. They were done in France or UK, in China, and now it's more and more clinical studies that show that extracellular vesicles are safe, feasible, and efficient in induction of anti-tumor response and in the treatment tumors. So same like with the cells, we can isolate cells and then isolate exosomes from them, or we can make these exosomes from milk or from grapes or other plants and load them with the drug and administer to the patient. So the most important, very similar to the cell-mediated drug delivery, it's the regulatory point. You need to think about potency, identity, that it would be the same formulation every time, reproducibility, purity and sterility, and storage conditions. But with storage conditions, actually it's easier because it's not cells. You can lyophilize them, store them, and then restore in the saline and administer when it's necessary. We use sometimes bioreactors. You seed cells on this filter, and then media goes through, and you can, over the several weeks, as long as the cell's growing, you can collect exosomes, take them into the big batches, and then characterize them all together, load, and work with them. So overall conclusions, exosomes are amazing, have amazing properties as a drug delivery vehicles, as with their own biological activity, and as carriers for potent therapeutic substances that can be delivered across impermeable barriers, and it could be used, but we need to work on it to work with this formulations. It's not all clear and easy, but it's very promising. So I used maybe 5 minutes more, but we started later, right?
Yes.
Okay.
Questions, please.
Yes, I've got one question. Maybe I didn't catch it. What are the problems of applying exosomes as a drug delivery?
Problems with the what?
Of applying exosomes as a drug delivery system.
So the problems are multiple, basically. If we will start from the first problem, which now on my almost last slide, I put, there's a lot of companies now that isolate significant large batches of exosomes, characterize them, and purify them. So first problem, just to get big quantities of these nanocarriers. For instance, for polymer-based nanoparticles, you can produce a bucket, I don't know, a lot of polymer-based nanoparticles, store them, and load them later. In this case, you need to have ability to isolate a large amounts of exosomes. The second problem is the loading, efficient loading of exosomes, because when you make nanoparticle-based formulation or liposome-based formulation, you can create these particles in the presence of the drug. So you basically creator of these nanocarriers, and you can mix them and then make these particles together with the drug. In case of exosome, it's already created, and you really do not want to screw up this beautiful surface and lipid content. So you have to be very careful to make holes and to load it efficiently, which is not easy because they are very stable and very stern. But on this other side, load it with a lot of drug. And the third problem is you want to keep them with low immunity. So you basically, the best, you take them from the cells of the same patient. Same like with the cell-mediated drug delivery, you need to think how many exosomes you can receive from the same patient and how can you do that. So for instance, when we work with monkeys, we isolate blood, white blood cells, and then isolate them from them with exosomes. How many exosomes you can this way, and how long you need to time you need to prepare this formulation. It's good when you can wait, like for Alzheimer and Parkinson. And what if it's glioma, brain tumor, which develops very fast? You need to administer it like yesterday. So there's the 3 main problems with the natural prepared, natural-based drug delivery system.
But there are some advantages also.
But advantages are huge, and we're not trying to make it for the diseases where it's easy to deliver. For instance, if you have a knee inflammation and you can inject your anti-inflammatory drug right into the knee, great. We will never propose to use exosomes for this treatment. But if you need to deliver a drug across blood-brain barrier, or specifically to metastasis or triple negative breast cancer. Here, you want to have this more complicated formulation that might work, because other formulation would not work, and it was shown that they're not working.
Thank you. Any other questions?
May I ask another one?
Absolutely.
You said that we can isolate exosomes from milk, yes? Or no?
Milk, yes, actually,
Yeah.
they already in clinical studies, this doxorubicin loaded exosomes from milk, yes.
And do they have bigger immune response of organisms, or in comparison with the exosomes which were isolated from exactly this organism?
Very good question. Very, very good and smart question. Yes, that's why I told you that I would not say that they have the same efficacy. Absolutely. But in this case, what these clinical studies, they give it perorally. So like you drink milk, so you can drink this formulation with doxorubicin perorally. And they have very good results. The clinical investigation have not finished yet, but they might have more immune response than like autologous exosomes. You're absolutely right. But can you imagine how many of them? So all this, cotton cheese and cheese production, they all have this liquid on top, and you can collect millions of these exosomes, and usually people just trash them. But you can use this production and have tons of these exosomes. So and for instance, grape also, it's interesting, but when people make this grapes, this wine and everything, right? There's a lot of exosomes over there. So it's just very useful because it's easy to collect huge amounts of exosomes, but they might be immunogenic. Yes.
Mm-hmm.
But for oral delivery, it's okay. Yeah, I think.
Yes.
Any questions? Is there any? No. Then thank you very much, Elena, for very interesting lecture.
Thank you for listening.
Thank you very much.
Thank you very much. Really interesting.
Mm-hmm.
Thank you.
Thank you, guys.
Thank you. Bye.
Bye.
Bye, everybody.
Bye.
I forgot.
Yeah,
Did you put plus to this chat?
Yes, I guess.
Yeah, we did.
Okay, thank you. Bye.
Bye.
Bye. Thank you.